So we are moving on into the metabolic strategies, and it's really cool. I love that topic. I think it's just super elegant and super cool. So we're going to start with broad definitions. We need to define what's anabolic, what's catabolic, because metabolism, definition of metabolism is kind of stupid. All reactions that happen in a cell, okay. So, anabolism, reactions of synthesis. That's the level that I want you to know. Catabolism is degradation. Okay. Now, another thing. Some reactions release energy, some require. Okay. So reactions that require energy are endergonic. Energy goes into the reaction. Reactions that release energy are exergonic. Question? What?
Okay. Sorry.
Okay. I'm going to give you examples. Absolutely. So let's think about a few reactions. We're going to start with super simple. Okay? Bless you. Here's the illustration. ATP is broken down to ADP and inorganic phosphate. In the opposite reaction, ATP is synthesized from ADP and inorganic phosphate. This reaction, the top one, is it anabolic or catabolic? Catabolic, yeah. I told you before, if I ask a question and don't assign the level of stupidity, it's 0 or 1. Now, when ATP is broken down, is energy released or not? ATP is the energy currency of the cell. Yes. So catabolic, exergonic, right? When you synthesize ATP, when you synthesize anabolic, do you need energy? Of course. So endergonic. When you synthesize proteins, anabolic, do you need energy? Sure. 2 ATPs per amino acid. Anabolic, endergonic. Generally speaking, reactions of synthesis are endergonic. They need energy. Reactions of breakdown of catabolism are exergonic. They produce energy. We good so far? Okay. We're going to leave it here, switch gears completely, and talk about two things that every living organism, including bacteria, need in order to grow. Energy and carbon, right? Where can you take energy? There are basically two energy sources. One is light, and another one is chemicals. If a microorganism can get energy from light, it's called phototroph. The root of the word troph generally refers to consumption of something, okay? So it's consuming light. The organism gets the energy from chemicals, then it is called chemotroph. So a photosynthetic cyanobacteria will be autotroph. The garden variety bacteria that we grow on the agar plates, they will be chemotrophs because they don't photosynthesize. They derive energy from chemicals. Now what about carbon sources? So carbon can be obtained from either inorganic or organic sources. So if source of the carbon is inorganic, CO2 or carbonate, you don't need to know the examples, like just inorganic. So basically, if a microorganism can take an inorganic carbon and make organic out of it, such microorganism is called autotroph. Okay? Good? And if microorganism has to get its carbon from organic sources, other organisms, either living or dead. Does that make sense? This organism is called heterotroph. Again, I'm gonna use humans as an example. Like, we eat food, right? We eat highly processed dead animals and plants. Make sense? Unless you like eating an apple straight from the tree without removing it. Okay? So we eat highly processed dead animals and plants. We are heterotrophs. Plants, first of all, they use energy of light, they phototrophs, and they take CO2 and convert it into sugars. They autotrophs. By combining these strategies, you get the full name for the organism based on their metabolic strategy. You should be able to match the description to the name. So if I tell you cyanobacteria use energy of light and they can convert CO2 into organic compounds, you tell me, oh, cyanobacteria are photoautotrophs. Make sense? Fungi, most of the bacteria, they will use energy from chemicals and carbon from organic sources, chemoheterotrophs. Bacteria that live near volcanic vents at the bottom of the ocean, there's not a lot of light, so they will use energy of chemicals, but they're able to convert inorganic carbonate or CO2 into organic compounds. So chemoautotrophs. Purple non-sulfur bacteria, they use energy of light but require organic compounds, photoheterotrophs. Does that make sense? I basically gave you like examples of the definitions. Clear? Okay. Now we're going to go back to the story how organisms generate energy, and to do so I have to. Okay. I have to say this. The whole story of energy transfer in the cell is about transfer of electrons. And I'm very confident in what I'm saying. So basically, you have compounds that have a lot of chemical energy in them in the form of electrons, like glucose. And somehow you try to strip those electrons from them and transfer them to somewhere else. So basically, you take electrons at this level of energy. Does that make sense? Okay? And you get them to this level. The difference is your energy yield. Am I clear? So it's all about transfer of electrons. We are going to talk about electrons because it's much easier to explain how this whole energy transfer happens. First, we need to define a couple of things regarding electrons and electron transfer and redox reactions. So here you have what I'm going to be talking about is purely an English language challenge. We're not talking about chemistry, we're talking about terms, okay, to use them properly. So you have chemical A and chemical B. Chemical A loses electrons to chemical B. Okay? We good? So when chemical A loses electrons, it is oxidized. When chemical B gains electrons, it is reduced. Does it make sense? Awesome. Seriously, thank you for the confirmation. Now, when B is reduced, what reduces B? chemical A. So chemical A reduces chemical B. And chemical B oxidizes chemical A. So by these functions, we can assign them names. A, in this particular situation, is a reductant. B is oxidant. Does that part make sense? That's just the terms, just the terms. So, since we figured that energy transfer in a cell is the transfer of electrons from one molecule to another molecule to another molecule to another molecule, what is the very first molecule that we think about when we think about energy? It's carbohydrates, or specifically glucose, okay? So our focus for the next probably 20 minutes will be a single reaction, probably the most famous reaction in metabolism. It's oxidation of glucose. Do you need to remember all the coefficients and stuff? No, it's not a chemistry class. Okay, so look, and this is important, look. You get a molecule, strip all electrons from that molecule and give those electrons to oxygen. Does that make sense? So if we kind of transpose what we have here to what we have here, the role of molecule A is assigned to glucose. The role of molecule B is assigned to oxygen. Okay? So electrons from glucose go to the oxygen. Glucose is oxidized. Oxygen is reduced. This is the catabolic reaction. Glucose catabolism. And as we mentioned before, catabolic reactions are usually exergonic. So as the result of this reaction, energy is produced. Does that make sense? And what this energy is used for? It is used to produce ATP. This statement that, mitochondria in eukaryotic cells or enzymes convert glucose in ATP drives me nuts. No, they don't. They convert glucose into water and CO2. That's it. Does that make sense? Good? Questions about this part? Now, in chemical world, that would mean what? You take glucose as a powder, put it in a chamber, fill it with oxygen, set it on fire. Glucose burns, literally burns, converting into CO2 and water. It doesn't happen in the cell. In the cell, the process of transferring electrons from glucose to oxygen is very multi-step process. Okay? So you have to carry electrons from one molecule to another, to another, to another. Are we clear? Electrons can't just move like ions, something. They have to be carried. So there are 3 electron carriers that we will talk about. I'm going to write 2 so far because 2 will only be relevant for the processes we're going to focus on. Deal? So those 2 electron carriers are NAD plus and FAD. When these carriers receive 2 electrons, what is the charge on the electrons here? Negative. You have to balance the charge, right? How you balance the negative charges? With positive charges. Huh? Absolutely, and this is where this confusion, and I'm going to call it confusion, is coming from. That, reduction is hard. No, reduction and oxidation is all about electrons, right? So you get NADH with another hydrogen hanging near and FADH or FADH, okay? Are you following me so far? So these molecules serve as some kind of a taxi for electrons. So electrons hop on, taxi is loaded, okay? They move electrons to wherever it's wherever it's needed. Then taxi can unload back. Okay, now looking at this terminological picture, tell me please, when say NAD gets electrons, what happens to NAD? It is when something is gaining electrons, it is reduced. So this form with electrons is called reduced form. Okay, both for NADH and FADH2. When NADH and FADH2 lose electrons, what happens to them? They're oxidized. So these are oxidized forms of the electron carriers. Okay? What I'm doing, I'm setting the stage. We are agreeing on the terms. We're introducing a couple of important players. We're looking at the whole process here because we're going to talk about something. We're going to talk about this process now in a lot of details. Okay? That makes sense?